Hi, this video is going to go over Minilab 10.1 from the textbook on page 281. How can an offspring's traits be predicted? A Punnett square can help predict ratios of dominant traits to recessive traits in the genotype of an offspring. This lab involves two parents who are both heterozygous for free earlobes, big E, which is dominant trait. The recessive trait is the attached earlobes, lowercase e. So in this problem, you had to go through the procedure here, and it basically helps you set up a Punnett square and then follow through and find the solutions at the bottom in the analysis section. So we'll go through the procedure here so you can set up your own Punnett squares and check your work, because you should have done that already. All right, first one is interesting. We don't really need to have the lab safety form for this because you didn't actually use any chemicals or any labware. So let's skip to part 2. Determine the gamete genotypes for this trait each parent contributes. So in genotypes, we know that they are heterozygous. And heterozygous is a carrier, but not showing the trait that is recessive. So we know that the genotype would have to be one capital letter and one lowercase. That makes them heterozygous. Now, if we were asked the phenotype of the parent would be free earlobe. You may not have noticed it before, but people have earlobes, so they can be attached or detached, and that is predicted through a single gene. And it's a dominant characteristic if it's free, and it's recessive if it's attached. So in order to have attached earlobes, you have to have received both recessive alleles from a parent. Part 3 says draw a Punnett square that has the same number of columns and the same number of rows as the number of alleles contributed for this trait by the gametes of the parents. So we will need a Punnett square with 4 equal boxes on the inside of the square. In this case, there was no distinction between what the male and female genetics are because they both have the same alleles. Because they're heterozygous, you get one capital allele and one lowercase outside of our Punnett square, top and bottom. Since this is the male symbol here, the symbol for Mars, and this is the symbol for Venus, this is the female, I made eggs and I made sperm. Now it also says write the alphabetical letter for each allele from one parent just above each column, and write the alphabetical letter for each allele from the other parent just to the left of each row, which we already did. Number 5, in the boxes with the table, write the genotypes of the offspring resulting from each combination of male and female alleles. So we have the first sperm and the first egg joining in the very top left box, and they both get 2 capital E's. That'd be a free. If we read that genotype, the type of phenotype we'd see, the outward trait would be free earlobe. We take the same egg, but we're going to join it with the second sperm, and we're going to get a capital E and a lowercase e, so a big E, little e. And again, it's still going to be a free earlobe because, just like their parents, they're heterozygous and they have free earlobes. If we take the first sperm and the last egg here, we get a capital E, lowercase. Doesn't matter which one's first because the large or the capital letter always overpowers because it's dominant, rule of dominance. And what we get is again free as a phenotype. So the genotype there is big E, little e. Phenotype is free. And the last one, our last sperm, our last egg, little e, little e. This is our individual that has attached earlobes. So we did one that's different. So one of the things we get to see here right away is that there was that hidden trait, right? Parents were carriers, but not showing the attached earlobe. But because they're both carriers, they have one 1/4 chance of showing that a child could be born with attached earlobes being different than the parents themselves. Again, you can sort of see this as a carrier skipping a generation. You can see how that could possibly happen with recessive alleles. All right, let's take a look at the analysis part. You'll have to do analysis on any of your genetics problems. The first one here says summarize a list of possible phenotypes that could occur. All right, so we have both free and attached earlobes are possible. Those are the two different phenotypes that we're looking at. Now, the key there is that it asks for phenotypes and not genotypes. Phenotypes are the traits that you're looking at. So free earlobes and attached earlobes are what we're looking for. Second analysis item is to evaluate what is the phenotypic ratio of the possible offspring and what is the genotypic ratio of the possible offspring. Let's start with the phenotypic. So the phenotypes, we know there's two. We know there's free and we know there's attached. All right, you got to look at our diagram here. All right, and we can see that for the frees, there's 1, 2, and 3. I've already labeled them. That's 3/4 or 75% are going to be free earlobes, and that means that to add up the 100%, you can see there is our single recessive individual, right? That's attached, 25% attached. All right. Now, the second part, it's asking us what is our genotype. Now, genotypes, we're looking at those symbols that we created to show what alleles were being inherited by probable offspring, right? Now, this doesn't mean that 4 people or 4 individuals are born. This is just percent chance, right? 25% chance of free earlobe, 25% chance of attached earlobe. Genotypes, now we're looking at what's the probability of having a homozygous dominant, which is capital E, capital E. What is the chance of having a heterozygous individual, a hybrid just like their parents, right? Carriers. And then what is the chance of having the recessive trait, meaning they inherited both of the recessive alleles. We can count the boxes. There's just one box that's big E, big E. So it's 25% chance of being homozygous dominant. Heterozygous is 50% because there's those 2 boxes, just like their parents, big E, little e. And then there's a 25% chance of being homozygous recessive because there's just that one box with little e, little e. So homozygous dominant and homozygous recessive both have the same alleles, just of the opposite type, right? All right, and that's pretty much all you have to do. When you're solving one of these probability problems, you must make sure you do your analysis and you look at your details inside the Punnett square to make sure you get all of your probabilities. And again, remember, this is not individuals that are born. These are probable outcomes. It could be that even though it's a 1/4 chance of being recessive, it's possible they will be recessive, even though their parents don't show that trait, right? Sometimes you hear it skips a generation. Well, that's what this is. It's possible for a trait to skip a generation because it's recessive, and it have to have both copies in order for it to be expressed.